All right, so hello everyone, a very warm greeting to one and all. We'll be starting with the webinar in just a few more minutes. And meanwhile, we are waiting. It would be great if the participants can introduce yourself in the chat box. Let us know from what is your education background. All right, so we have Stacey. I hope I'm pronouncing your name correct. Otherwise, please feel free to correct me. And he says he is from MSc Computer Science. That's pretty interesting. Can you also let us know what made you interested to learn about bioinformatics, your transition from computer science to bioinformatics? And we also have Maureen. She is an MSc Bioinformatics and Molecular Biology student from Kenya. Yes, Maureen. Hello, nice to meet you. And Sadik is pursuing Master of Engineering in Information and Electronics Engineering. We do have a diverse range of students amongst us. I'm curious to know about your transition as well. What made you interested to learn about bioinformatics? Let us know in the chat. All right, I can see that few more participants have joined, and few of our research fellows have also joined. So just to give you an overview, in today's webinar, we will just be introducing the participants to the research fellowship program. And we're also joined by our former research fellows who will be presenting their project. And it's going to be an interactive session where you can ask queries about the program and learn more about it. And we do have a couple of responses in the chat. Cecil says he wants to help fight complex diseases like cancer with his computational skill. That is pretty inspiring. And Maureen says the ability to be able to analyze genetic makeup of an organism to understand the reason why and give solution to an existing phenomena. That's also pretty interesting to know. Thank you so much, Maureen and Cecil. Sadik has also shared. Currently, he is working in anti-cancer peptide classification using a computational model, but he's not sufficient about this and is working on generating an anti-cancer peptides using a generative adversarial network. So here comes a bridge to learn molecular mechanism of peptide to different cancer cells. And besides this, I also want to learn molecular talking. All right, thank you so much for that description. Do you have you also learned about the research fellowship program, or are you new to this? Maureen, I know you are already a research fellow with us. What about the rest of you, Sadik and Sisig? All right, Sisig, he has already signed up for the program. Yes, I just got to know about that. And Sadik, he is working at John Book National University as a researcher. All right. Thank you so much, everyone, for introducing yourselves and letting us know about your research interest. And meanwhile, the participants, rest of the participants can also let us know about your educational background and research interest. And in the meantime, other participants will be joining us. And without further delay, let's start with today's webinar. So hello, everyone. A very warm greeting to one and all. My name is Sri Gauri Krishnakumar, and I would like to welcome you all for the webinar on OmicsLogic, Bioinformatics, and Data Science Research Fellowship Program. So those of you who are joining us for the very first time, the Research Fellowship Program has been designed to help young researchers and students take advantage of the bioinformatic resources for the analysis of complex life science data and become well-versed in bioinformatics. Research fellows participate in cutting-edge bioinformatics research led by expert mentors, and the fellowship program will offer a combination of online resources and mentor guidance to prepare you and help you complete a bioinformatics project. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of our mission, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by over 24,000 participants from 187 countries in over 300 workshops and in different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, and data science. And as a part of the training, you will have access to the OmicsLogic resources, which takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path of the Bloom's taxonomy pyramid. And this also comes with an access to the OmicsLogic courses, which includes courses on bioinformatics, precision medicine, multiomics, that consist of genomics, transcriptomics, metagenomics, and epigenomics, data science that makes use of bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. The OmicsLogic platform also gives options for analyzing a given dataset, and these datasets and associated publications can be used in several ways. That is, you can learn bioinformatics without any coding at all. And for those interested in opting the non-code pathway, they will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines, as well as data management and analysis cloud infrastructure to run the bioinformatic pipeline. And different stages of analysis are performed in different sections on this multi-omics platform. And for those interested in learning to analyze data through coding, we provide the OmicsLogic Code Playground, which will help you develop coding skills while learning popular packages in R programming and Python to visualize, annotate, and analyze complex patterns in biological data. So now to view the various courses, example projects, student projects, you need to first sign up on the OmicsLogic Learn portal. So let me walk you through on how you can do that. So those of you who have not signed up on the portal, I'm sharing the link for signing up in the chat box. Please visit the link and I'll walk you through on how you can sign up. So if you're signing up for the very first time, when you visit the link, you'll be able to see the create an account option. Click on that. And when you click on that, you will be able to create an account by entering your name, providing an email and a password. Or you can also sign up using any of your social media handles. It could be a Google account, Facebook account, Apple account, GitHub account, or even Twitter. And for those participants who have already an account on the portal, you can simply click on the login now option and simply enter the existing email ID and password and just sign. So that was all about how you can sign up. And with that, I'll pause for a couple of seconds for the participants to sign up. And once you have, please let me know in the chat and I'll proceed ahead. If you already have an existing account on the portal, you can put a done in the chat box so that I know you're good to go. And if you're facing any sort of technical issues, you can let us know that as well in the chat box and we'll be happy to look into it. All right, we do have a couple of responses in the chat that they have signed up. All right, since I can see that some of you have already signed up, so I'm just going to proceed ahead, but along the way, if you're having any queries, you can let us know and we'll be happy to help. So since I already have an account on the portal, I'm just going to click on sign in. And once you sign in, the very first page that you will be seeing is the courses tab. So under the courses page, when you scroll down below, you will see a search bar that says looking for a specific course. So here, if you are to enter any area of your research interest, so for example, if you're interested to learn about transcriptomics, then that will show you the various courses and example projects that makes use of the concept on transcriptomics. And as you can see, there are several courses, one on transcriptomics, then single-cell transcriptomics, then analyzing transcriptomics data using R programming and Python, and several other example projects as well. So you can simply enter any of the keywords that you're interested to learn more about, and it will show you all. So let's say I can see that a few of the participants are interested in oncology. When I enter the term oncology, it shows me several example projects that one can learn more. So that was all about how you can navigate the courses page. Now, the very first thing that you have to do once you sign up is to set up your account. To be able to do that, you can click on the blue bar that is welcome back with your email ID. When you click on that tab, it will directly take you to your profile settings. Here you will see a button that says profile. When you click on that, it will enable you to edit the features that you are required to update on your account. So what are these features? First of all, make sure that your name is in proper capitalization, because once you complete the courses, the certificates will be automatically generated. So it is important that your name is in proper capitalization. And the second thing is to upload a profile image as well. In case you're not comfortable adding your own image, you can add a university's logo or an avatar for the profile image. And the next section that you need to update is your bio, wherein you can briefly mention your educational background and your research interest. And finally, after you link your social media handles, just like in my case, I've linked my LinkedIn account and my Rock ID, your profile will be complete and you'll be there to go. And now you will also see several tabs next to your profile, that is your activity tab, wherein the program mentor and the community manager, they will be tracking your learning progress from this part of the tab. And here under the courses tab, you'll be able to keep a track of all the courses that you're currently completing, and it also shows the coursework progress that you're making. And finally, once you complete a research project and publish it on the portal, you'll be able to get the projects under the projects tab. Finally, if you're enrolling for any of the other programs, then you'll be able to see the programs under the programs tab. And finally, once you complete the required coursework with the associated assignments and quizzes, you will be able to receive all of the certificates under the certificates tab. So that was all about, I wanted to tell you about setting up your account. Now, another important tab that I need to show you is the projects tab, as you can see here. When you click on this, you can check out all of the student projects that have been compiled and put together. And all of these are research projects, and a lot of them are presently working on their research publication or working on poster presentation competitions. And all of these project topics range from oncology, infectious diseases, machine learning, data science, space omics, and so on. So the participants can have a look at all of these at your own pace. There are tags provided on the right, and you can click on any of these and it will show you the associated projects. So that was all about navigating through the OmicsLogic Learn portal, and this is the portal that you will be making use of as a part of your program to complete your coursework and required assignment. So that was all about I wanted to tell you. And with that, now I would like to invite our former research fellows, Mr. Ojasvi Datta and Shubham Kumar, to present their research project on effect of microgravity on human-induced pluripotent stem cells. So with that, the stage is all yours.
Thank you, Shrigori. Hello all. I'll just take a minute to share my screen. I guess Shubham has also joined the session. Yeah, I can see him.
Yes.
Can you hear me?
Yes, I can hear you.
So, is my screen visible?
Yes, it is not in full screen mode. Can you go to that?
Yeah, I'll just do it.
Yes, now we can see your screen. Thank you.
All right. So, hello all once again. So today I and Shubham will be presenting our research project, which we have completed with Pine Biotech for our dissertation. So the title of our project is Human-Induced Pluripotent Stem Cell-Derived Cardiomyocytes Exhibits Delayed Recovery Rate Due to Stress Induced by Spaceflight. And we completed this in the domain of space omics using transcriptomics coursework and introduction to bioinformatics. So I'll just go through once from the introduction, starting with astrobiology and microgravity. So the study of origins, evolution, distribution, and future of life in the universe is known as astrobiology. It is an interdisciplinary field which demands a thorough knowledge of biological, planetary, and cosmic events. Microgravity is the most important influence on the human organism in space, and the human body undergoes dramatic changes during long-term spaceflight. The long-term exposure of microgravity by spaceflight and bed rest on the ground result in severe osteoporosis and muscle atrophy and neurological disorders. So, changing in the structure and mass of the heart, we decided to work in studying the cardiac functions, cardiac arrhythmia, and lower body vascular regulation in this whole research project. So talking about the effects on the human body by space travel, as you can see in this pictorial representation, the human body is adversely affected by microgravity in all terms, like in veins, platelets, brain. Radiation also causes harm to DNA and muscle loss, muscle strength, and also it affects the cardiac related functions. So what is pluripotent stem cell? Human pluripotent stem cells including embryonic stem cells and induced pluripotent stem cells, have the capability to self-renew indefinitely and to differentiate into derivatives of the three germ layers, ectoderm, mesoderm, and endoderm. In 2006, the first induced pluripotent stem cell were generated by reprogramming skin cells. Also, they are derived from somatic cells and has enabled the development of personalized medical strategies and patient-specific disease models. Pluripotent stem cells have the ability to like self generate or renew themselves. So that is why they can be differentiated from heart to many other uh, related functions in the human body. So in this project, we use these steps as a method for completing the research. First was to retrieve the dataset from NCBI GEO. Then we did the gene quantification and normalization of raw data. The third step was to do principal component analysis, which is an unsupervised machine learning analysis. Then we did the differential gene expression analysis, followed by KEGG and geo pathway analysis. And then we did some manual curation for cardiac and muscle related activities to find the major genes and pathways which are causing the harm to the cardiac related muscle functionality in human. So this is our GUSS number for the dataset, which we have used for our study. The title of the study is Effects of Spaceflight on Human-Induced Pluripotent Stem Cell-Derived Cardiomyocyte Structure and Function. So in this study, the authors tried to maintain the study on the contractility of muscles, and they did some study on cardiac-related muscle pathway analysis as well, but somewhat they like put more efficient analysis on the cardiac muscle contractility and metabolic related pathways. So the microgravities, as we know that it induces specialized cells and stem cells, the spaceflight condition. So we decided to work further on the human cardiac and muscle activity. And in a couple of studies, it was discovered that induced pluripotent stem cells transformed into cardiomyocytes may restore the contractility of the heart. And the researcher decided to replicate the recent work on mice using human-induced pluripotent stem cells. It was primarily focused on the impact of calcium handling and the impact of heart contractility. And then we decided to work further on this heart-related analysis. So this is the pictorial representation of the GEO dataset page and sample characteristics. The whole study was done in the period of 5.5 weeks, where 4.5 weeks the samples were kept in the microgravity exposure, and then they were kept for a rest period and for the 1 week of time. So there are a total number of 18 samples in our dataset, 6 each in different conditions, in-flight, post-flight, and ground control conditions. We created 2 different groups based on the samples in the dataset. One is in-flight versus ground, and then post-flight versus ground condition. So as the data was normalized on GEO, we collected the raw file from SRA, and then accordingly analysis was performed using different pipelines. So we used RNA-seq analysis, quantile normalization, PCA, and DSEQ. So what is RNA-seq analysis? So RNA-seq is a technique that can examine the quantity and sequences of RNA in a sample using NGS. It analyzes the transcriptome, indicating which of the genes encoded in our DNA are turned on or off and to what extent. Then we did the quantile normalization to standardize the data obtained following gene quantification, because it had various void spaces and null values. And in the same pipeline, we used the PCA component to visualize the data. So here's the pictorial representation of our result of PCA. So it denoted that we found that the clustering of sample 2 and sample 5 in both the conditions, ground versus flight and ground versus post flight, are same. The difference we observed was that in ground versus post flight condition, this one, uh, due to the microgravity stress, the togetherness of the samples was bit deviated, and as compared to ground versus flight results. So the whole pictorial representation of this species states that the samples in all the areas lies in the same function like G1, G4, G1, G4, and uh, in post-flight PF3, PF4, PF1. But here you can see that in flight condition, we only had F1, F4, and in the above symmetry, we had G6, G3, F3, and F6. But here in this one, the F2, F5, G2, G5, G2, G5, PF, P2, and PF5 samples are deviated than the flight condition. So now I would like Shubham to continue from the DSEC analysis part. Shubham, can you just take it over?
Yeah, thank you so much, Ojasvi. Could you confirm if my voice is audible properly?
Yes, your voice is audible.
All right. So, hello everyone, and thank you, Jasvi, for such a great explanation. So, like moving forward, after the PCA analysis, we perform the differential gene expression to determine the level of expression, that whether they are upregulated or downregulated in the cell. So, we perform the DSEC2 analysis using the T-band for server. You can see the pipeline which we have used and the parameters we have set up for the for our differential gene expression analysis. Now here next we can see that the uh, like in the next slide we can see that the heat map of our data set for the both the ground versus flight and both and for the ground versus post flight condition. And we also the togetherness of the closeness of the sample which we can observe from here, like for flight 1 and flight 4, as well as for the ground 1 and ground 4, which we have like quite similar with the PCA results. And we along with the DSEC analysis, we did the pathway analysis using KEG and GEO. So from that we found the dot plot for the KEG analysis, as you can see on the screen, and for the GEO analysis. So these are the dot plots which, from where we can see that some of the pathways like ribosome and other related to the like spliceosome, and some of them related to the neurodegenerative diseases. But because these were the human induced cardiomyocytes, and the previous study was done on the cardiac study, so we also focused to study our uh, like research work to be on cardiac and muscle related activities. So when we did that, we found that for the geo analysis we found approximately 591, and for the post flight condition we found approx 1200 genes. So you can see that the number of genes in the post flight condition, that is after the microgravital stress, the number of pathways we got, they are approximately doubled. So similarly, this was observed in the KEG analysis. So as I said that we performed for the cardiac activities and for the muscle activity. So for the cardiac, you can see that the for the ground versus flight condition, we found only 9 pathways which were highly enriched, and for the ground versus post flight, we found approximately 66 pathways, which was around 5 times than the flight condition. And similarly for the muscle related pathways, we have found that you can see the numbers like 29 pathways in ground versus flight, and 20 and 47 in post flight condition. But for the KEG, we didn't found any result of in the flight condition, but in the post flight we can see that one pathway is highly enriched. So and based on that we found that some of, when comparing all these data, we found two unique pathways which were either suppressed or activated in one of the conditions, either in ground versus flight or either in ground versus post flight condition. So the first unique pathway which we found was the signaling pathway regulating the pluripotency of the stem cells. And using the disjoint results, we got to know that these pathways are somehow responsible for the embryonal carcinoma or for various like cancers like stomach carcinoma and malignant neoplasm of the stomach. And the genes which are highly enriched for these particular pathways, as you can see on the screen, is hox b1 and fgfr4 and hand1, all these things. Now like here you can see the pictorial representation of the complete signaling pathway regulating the pluripotency of stem cells and the genes which are highly enriched. So these genes are present in the our data set as in our results, as well as the pathways which is like responsible for the stem cell activity. Now, the second unique pathway which we found in the muscle, which we found related to the muscle activity, was vascular smooth muscle contraction pathway, which was activated after the microgravity stress. So, this pathway might be responsible for the hypertension or the hypersensitive diseases, or somehow related to the chronic heart failure. And the genes responsible for these pathways are like ADCY6 and all that. So here, yes, you can see the pictorial representation, and you can see that how some of the genes are responsible, like MLCK, then CAM, are responsible for the pathway functioning. Now, what we found that authors were focused on studying the calcium activity of heart relate or the heart relaxation or the contractility. But what we found is that this particular pathway, like the vascular smooth muscle path contraction, was totally dependent upon the calcium for its functioning. So these were all the unique findings from our data set and from our research work. Now, coming to the conclusion part, what we conclude from our research work is that the pathways enriched, they are statistically relevant but not biologically relevant, because they were representing the cardiomyocytes, and they are responsible for the neurodegenerative diseases also. Now, the another conclusion we made was the cardiac pathways were not significantly changed in the terms of in-flight and the post-flight condition, comparing the gene level. And because of the stress, we observed the muscle contraction, like the pathway which we have observed. And one more thing, what we found is that the 1 week of rest for the recovery of cells is not sufficient for the cells to perform the like normal function or to get in a normal state. And in the ground versus flight and in the ground versus post flight, as we have as we have observed on the plots, that the enriched pathways are doubled in both KEG and GACA. And the two unique pathways which we have found in our analysis, the signaling pathway regulating the pluripotency of stem cells was observed in the ground versus flight condition, but was suppressed after the flight condition, that it was not found in post flight condition. And the pathway related to the muscle was activated in the post flight condition because of the stress observed in the microgravity conditions. So this was all about our research project work. Thank you.
All right. Thank you so much, Shubham and Ojasvi. And just to wrap up the presentation, could you please also let us know, you have won the poster presentation competition twice, right? So do you have any advice for those participants who are interested to join for the upcoming symposium on how they can ace their poster as well?
Yes, so I can just show the poster which we have made for the last symposium. Let me share my screen again. Can you see it?
No, not yet.
Yeah. Now, is it visible?
It started sharing and then it just stopped.
Um, it says the window is Closed. Can you see it now?
No, the screen is not being shared, it seems. Yes, now it is showing started screen sharing. Yes, now we can see.
Okay. So like for the poster presentation, we have just like included all our major work which we have done and the unique pathways, like the unique findings from our project. So uh, we have captured some of the information from the introduction, the major one, which we thought that it is important for the uh, judges and other researchers to like showcase. And then we decided to keep some of the major methods which we have used, like DSEC2 analysis, and what number of samples we have used and what group we have created for our analysis. Then we also stated some of the like basic values which we have further kept for our data to be sorted accordingly. And then in result and the conclusion part, we have kept all the key findings which we have like found in our all the research work which we did for this specific project. So my suggestion is to just keep it really specific to what key findings you have in your analysis, and not to provide any further supplementary data or the findings which is not relevant or maybe uh, maybe available in the author's paper. So you just have to give the unique uh, key findings in your like poster, and it should be like in a better representation so that the people can watch it um, with their naked eyes. So it's like an 84 percentage. If I will keep it in 100, then it will be more visible for all of you, I guess. So it is easily uh, like readable, and you can analyze like what we have found in our project for it.
All right. Thank you so much, Ojasvi and Shubham, for joining with us and sharing your research work. So now let me share my screen. So far we discussed about one of the research outcome was that students participating in poster presentation competition. But apart from that, there are several other outcomes that we have noticed as a part of students completing the research fellowship program. So let's just explore all of that one by one. So to begin with, most of the students, they are focused on publishing their research work. For instance, Rutvi Vaja, she is a biomedical science graduate student who worked on assessing the underlying reason for the development of gray matter lesions in multiple sclerosis patients. Further, she went on to publish her work in Immunogenetics, which is an open access journal that focuses on genetic research areas of autoimmune disorders. And another interesting student publication is that of Subanta De. He is a genetic engineering student, and he studied the effect of malaria vaccine, and he tried to understand the molecular mechanisms underlying the protection that the vaccine offers. And he carried out all of this using the transcriptomics datasets. Further, he went on to publish his work in Genomics and Informatics, which is an official journal of the Korea Genome Organization. Presently, we also have several other students just like them who are working on their publication. And for instance, Nivedita Arambam, she's a biotechnology graduate student. She's working on an oncology related research project, and it tries to understand the after effects of cancer treatment in patients. And like her, we are looking forward to several other publications from our current student. Further, You also have the opportunity to participate in poster presentation competition, just like Ojasvi and Shubham did, as a part of the OmicsLogic Research Symposium to win exciting cash prizes. And this symposium is an international symposium that brings together experts from different parts of the world, such as Dr. Gareth Rubble, who's a staff scientist at Lawrence Livermore National Laboratory, and Dr. Joseph Moore, and among so many others, experts who join as guest speakers and judges. And the students come forward and they present their research work in the post presentation competition. So that is another outcome that students have exhibited. And the student projects are not just limited to graduates and doctoral students or faculties or researchers. For instance, a very simple example is that of Andre Moadano Sosa. He's a high school student and who won a neuroscience project at the Regeneron International Science and Engineering Fair, for which he won the Potamkin Prize and the fourth award in biomedical and health sciences category. And this program can also be integrated as a part of the existing university curriculum. For instance, we offer the machine learning for biomedical data course, which is integrated into the systems medicine graduate program at Georgetown University. And this resulted in students completing their research project on a diverse range of topics, where the central focus was on machine learning. And these topics range from infectious disease, metabolic disorder, neuroscience, psychiatric disorder, oncology, and so on. So all of these projects are presently visible on the OmicsLogic Learn portal, which I showed you just while I was explaining about navigating to the portal. And these projects can also be showcased for higher studies and international university applications, like some of our research fellows did. For example, Dilara Daikin, who got accepted to the Erasmus IMBRC program after completing the research project. And Dhruv Mehra, he got accepted to the Precision Medicine graduate program at Manchester University. And Anusha, who recently got accepted to a metagenomics PhD program in university in Sweden. And faculties and researchers have also collaborated through our programs to publish their research work. And our latest collaboration has been with a team of researchers at Noguchi Memorial Institute for Medical Research at the University of Ghana to carry out a computational-based study to understand the variants associated with sulfadoxin resistance in Plasmodium falciparum. And it's just not limited to scientific related outcomes. It is also possible to pursue an entrepreneurship and business innovation track that, for example, like a team of industrial researchers from Biologic Support Services, a biotechnology company in Nigeria did. Their aim was to improve the fertility of women in Nigeria and improve their chances of carrying their babies to term by developing a test kit. So as part of it, they carried out a metagenomics research project. And the entrepreneurship track is not only pursued by researchers, but by students as well. For instance, a group of students from undergraduate and postgraduate program from VIT, they worked on developing an early detection kit of neurological disorders with a specific focus on Alzheimer's disease. So all of these are just very inspirational research projects that have been carried out from a diverse range of by students from a diverse range of backgrounds on a diverse range of topics. So all of these can be explored and carried out by all of us, just like these students. So that is all about that I wanted to talk about in this presentation. So now to learn more about the Research Fellowship Program, I would like to pass on the stage to my colleague, Ms. Parshthar, who will be explaining about how you can enroll for the program, what is the registration process, and how it is carried out. So over to you.
Thank you so much, Shri Gauri. And it was a very wonderful presentation, Ojasvi and Shubham. Thank you for explaining about your research project in a brief and crisp manner. So let me share my screen and guide you all through the different type of specialization tracks which we have and on which you all can work on depending upon your research interest. So as Shri Gauri explained in the beginning, that this, like the research fellowship program, has been designed for the young researchers, for biotechnologists, for biologists who want to take advantage of the bioinformatics resources for analysis of complex life science data and become well versed in bioinformatics. So, this is the right place and this is the right program in which you can enroll and further carry out the analysis. So, in OmicsLogic, so what we offer, so OmicsLogic offers high grade training and research tools for hands on analysis for various research fields, which include analysis of big data belonging to multiomic fields like genomics, like transcriptomics, like epigenomics, and metagenomics. Then there are further research specialization tracks for omics data analysis, like infectious diseases, which you can see on my screen, precision medicine, precision oncology, then neurosciences, space omics, metabolic disorders, agri-bioinformatics, immunology, cheminformatics, etc. We also aim to utilize data science techniques, programming languages like R and Python for bioinformatics, then some machine learning methods, bioinformatic tools and softwares, and AI based training platform, that is the T Bioinfo platform, of which you might have seen in the presentation of Ojasvi's and Shubham, the pipelines, etc. they ran. And so this is a platform which is being, designed by the Tauber Bioinformatics Research Center, Israel. So now what I'll show you. So here are some, like the below are some listed, here on my screen are some listed areas of research for various fields that OmicsLogic mentors are working on and are open for all to carry out independent or group research projects and able to go forward for a publication in their prospective or respective fields. So the very first specialization track is the infectious diseases, and here below are the mentioned omic data analysis in which one can work on. So if we talk about genomic data analysis, so we have listed out few of the experimental design on which you can work on. So first is comparing DNA sequencing using phylogenetic analysis, MSA, that is multiple sequence alignment. You can visualize and filtering alignments. Then the second is the transcriptomic data analysis, where you can identify the signature significantly different under therapeutic, drug treatment, and untreated conditions also. Then for the epigenomics data analysis, DNA methylation analysis, DNA protein interaction analysis. Then for metagenomic data analysis, you can analyze the microbiome of data and species diversity. You can determine the relationship between different communities, etc. So here are some project examples which we have listed on infectious diseases. And when you'll click on these images, you will be redirected to the respected research project. So one is on SARS-CoV-2 genomics. Again, the next is on malaria infection, then SF9 cells, biologics, Ebola virus epidemic, etc. So the next specialization track on which we are working on oncology, and it's right now in quite, it's trending, it's in hype. And below are the listed omics data analysis in which one can work on. So the first is transcriptomic data analysis, where you can where you can do downstream analysis of transcriptomic data, visualize to statistic analysis of differentially expressed gene, identification of transcriptomic signatures. When it comes to machine learning model, you can use regression and vector regression analysis. You can identify feature importance and interpretable ML modeling. Then we have genomic data analysis. We can integrate multi-omics integration also, where we can integrate multi-omics data to maximize information content to study cancer biology. Then we can, work on epigenomic data analysis, DNA methylation analysis, DNA protein interaction analysis. Then there is another aspect, that is structural biology and cheminformatics. So when it comes to structural biology and cheminformatics, we will be understanding the protein structural analysis, structural functional implications of prominent genomic variants, how we can explore cheminformatics strategy so as to model and screen and to improve the drug discovery results, exploring small molecules library based on structural similarity for candidate lead generation, and then comparing and contrasting their effectiveness through molecular docking and investigation. So again, here are some project examples which have been listed below for oncology, that is cancer, macrophages, TCGA, liver cancer risk, PDX tumor microenvironment, etc. And the third specialization track in which one can work is precision medicine. So in precision medicine, you can work on transcriptomic data analysis, clinical data analysis, epigenomic, genomic data analysis. And this is the example. This is the example research project which we have uploaded on OmicsLogic Learn portal. And if you can click on, you will have a glimpse of the what all steps or what the strategy which we have followed so as to complete the research project. And the next one is the agri bioinformatics. So in agri bioinformatics, we can analyze genomic data, we can analyze metagenomic as well as transcriptomic data. So we can study how distinct genetic profiles are associated with the condition, for example, drought resistance. And there is an associated example research project to it, that is potato drought resistance, gene expression of drought stress. Then when it comes to metagenomic data analysis, we can analyze the microbiome composition and species diversity. We can we can see the processing of 16S rRNA data. We can visualize the OTUs, operational taxonomic units, abundance tables can be seen for diversities. We can explore species richness through phylogenetic analysis, etc. And then in transcriptomic data analysis, we can again identify gene expression levels, interpreting results using statistical methods such as p-values, false discovery rates, etc. So moving ahead, then we have neurosciences. So in neurosciences, again, transcriptomic data could be analyzed. So what we can do is we can identify variability in differential expressed genes across the data set. And there is a example data sets in projects which have been linked over here. You can definitely open up this link and go through with it. And then we have an associated example research project again on the Learn portal, which is Alzheimer's disease patient pathology, differentially expressed genes and pathways. So you can take a full-fledged comprehensive look on this research project, that how it is, how we have done it, how our OmicsLogic mentors and team have completed and published it on our portal. Then next is metabolic disorders, where mainly we are working on diabetes. And in this we are working on transcriptomic data analysis. We are basically comparing the kidney transcriptomic profiles for early and advanced diabetic nephropathy, which reveals potential new mechanism for disease progression. And then finding RNA genes differentially regulated between the two conditions in order to identify the novel target, so as to find the cure, the treatment further. Then the space omics, again it is we can analyze the data in transcriptomic, then epigenomic, metagenomics. And today you have already learned, and you have already heard Ojasvi and Shubham explaining about the altogether new omic field, that is space omics. And these are below mentioned are the data analysis types which you can work on. So transcriptomic data is where they can find a differentially expressed genes, looking for significant altered pathways. Then analyze of RNA-seq data of organisms such as mice, zebrafish, microbes, etc. Then when it comes to epigenomics data analysis, then again we can talk about the bisulfite-seq, the chip-seq data organism in space, studying about their epigenetic changes in gut microbiome, etc. And then taking a look at the essential pathways such as WNTP53 and DNA damage response. So these were the specialization tracks which we are working on. So these all specialization track can be integrated with machine learning aspect or any of the programming languages like R or Python for the further interpretation for the novel finding. So when I scroll down, you can see here the research projects of are research fellows, and these all research projects are on different specialization areas. For example, some are on oncology, some is on space omics, some is on neurosciences, some is on precision medicine, etc. So you can take a thorough look on this page, and you can decide in which particular research area you are interested in and how you would like to take it further and develop your own independent research project and then publish into a research publication in a good peer reviewed research journal. So here are the glimpse of our some expert talks related to different specialization tracks, which you can see, and it's available on our YouTube channel as well. So it is again on different specialization track. For example, the first one is on omics in virology. So it is given by Dr. Rahul, which is a professor in Department of Microbiology and Immunology from University of California. Then we have a very good talk on molecular and cell biology and immunopathogenesis of coronaviruses from Dr. Gus Koselis. He's a president of NAPI USA. Then again, mathematical modeling of the single-stranded RNA virus by Dr. Leonid Brodsky, who is director of Tauber Bioinformatics Research Center, Israel. And then again, a talk on omics virology, system medicine, interpretable machine learning model for precision oncology, and so on. So, like there are multiple options for you to take a look on and decide in which particular specialization track you are interested and how you would like to integrate maybe our programming, Python programming, data science, machine learning aspects with all of these specialization track and come up with a research idea. So now you must be, thinking how you can enroll in research fellowship program. What is the program structure? What are the different resources which one could have access to, and then the fee. So to explain, so this research fellowship program, it's divided into three phases, or we can say three stages. And the three stages are: the first stage is to train you. So the first stage is training part. The second stage is your analysis part, where you need to, find an impactful research topic, properly prepare a methodology which you will following so as to come up with the results part. And the third stage is to present your analysis so as to get the expert feedback. So when it comes to training, we will be training you according to your research interest. We will be quantifying your skills in omics data, analysis, processing, and visualization and interpretation things. So, for example, you are interested in genomics. We will be providing you with that kind of material which will increase your theoretical as well as conceptual knowledge related to genomic data analysis. So some of the topics which we'll be covering in the training stage would be introduction to bioinformatics and basics of data in life sciences, so that you will get a glimpse of what is bioinformatics, what are the basic terminologies which we'll be using in bioinformatics, how bioinformatics can be used in healthcare industry, how bioinformatics is transforming translational research, etc. And then there would be some lessons on basics of data in life sciences, like what is central dogma, what is transcriptomics, what is genomics, what is metagenomics, what is epigenomics, proteomics, etc. So these all data types, omics data types would also be covered. Then data science and machine learning for non-engineers and biologists, and then R and Python programming and in-depth analytics for complex omics data. So these are topics we'll be covering. Again, we'll be covering those. We will be covering according to your research interest. So as I said, if someone is interested in any of the omic data type, so we'll be focusing. We will be providing that material, that particular resources, so that they get they have enough material so as to increase their conceptual and theoretical knowledge, which will eventually help them out so as to find an impactful research topic, to do proper literature review, to do a proper find out a proper data set on which they can work on, prepare their full methodology, and then do the analysis and interpretation afterwards. So the second stage is find an impactful research topic, where you will learn to perform literature review and plan out your project. So there are few steps few points which you need to keep in mind when you are in second stage of your research fellowship program. The first one is to make sure that your research question is compelling to those already in the field. So obviously you will be, you will be doing performing some literature review, and you will come up with some papers which will fascinate you, and you want to do some kind of similar work. But obviously those who have already done on that particular same topic, so you are not working on a novel finding, so it's almost similar. So you need to be extraordinary. Your research question needs to be more compelling than those who are already in the field. Then the second is set specific parameters to answer the research question conclusively. The third is find data sets that fit your objectives and explore them for batch effects or bias. Get feedback from our expert mentors on your topic and analysis proposal. So whatever data set you will finalize, whatever literature review you will be doing, whatever methodology which you'll prepare to analyze your data set, to do the interpretation, to get the results, etc., these all steps would be mentor guided. So our expert mentors would be there to guide you on each and every step, so that you come up on a proper and constructive conclusion. Now, the third step is present your analysis for expert feedback. So you need to develop a complete project ready for expert review and feedback. So once your project is prepared, it becomes the proof that you have understood the topic and analyzed data correctly. Expert feedback and peer review will identify gaps and omissions you can address before sharing the final results with the scientific community and the world. So once your project is done, you need to jot it down in a proper manuscript, scientific manuscript, and do the editings. So that is done by the expert feedback, and our mentors will eventually help you out in those, like in correcting your mistakes in your writing patterns, and they'll identify those gaps and will fill up those voids. And after your research project is done, and what are the potential outcomes? So one of the potential outcome is expert feedback and overall evaluation of your project quality, innovation and analysis execution. Second is the poster presentation at the research fellowship meeting or a research symposium. So this month on 30 and 31st, we are organizing a research symposium, and we are inviting each and every participant out there, if they have done any of like if they have done any research project with their PIs or with their faculty or their university or colleges, they can come and they can present their work on in Omics Research Symposium in the poster presentation competition and win some exciting cash prizes also. Those who are interested, they can ping their email address in the chat box, and we'll convey and we'll provide you with the details of the poster presentation competition and research symposium as well. Now, the third is path to collaboration with expert mentors or faculty at the partner universities. And then the last one is path to a full publication. So you can get your research paper published also, as Sri Gauri has given you a number of example in her presentation of those who have already published their paper and those who are working on getting their paper published, their research paper, their research work published. So definitely there is a path to a full publication. Now this was a structure which we will follow. Now coming up on the tenures and the resources. So there are 4 different tenures in which one can join. One is 45 days, 60 days, 90 days, and then there is another one that is 180 days, that is 6 months. Then we have common resources for all the 4 tenures, that is asynchronous courses, hands-on assignments, group sessions, program course access, OmicsLogic program access, educational cloud pipelines, online technical support, online mentor support. Then if you get stuck somewhere, there is an option of one-on-one mentor guidance as well. So these are the resources which are common in all the tenures, but there are outcomes. There is difference between the outcomes. So if you'll enroll for 45 days, the outcome is a literature review and a intermediate level of project with the project report. Then in 60 days, You are applicable for a literature review, intermediate level of analysis, then and a project report for that intermediate level of analysis, and then if it's ready, you can present in a research, or you can present in research symposium as well. In 90 days, there would be a full-fledged research project, and this particular project would be, applicable for the research paper publication as well, and similar for the 180 days also. So these are some resources, outcomes, and the tenures which one can take a look. And then when you scroll down, you will find there are three different payment checkout links for research fellowship program: 45 days, 60 days, 90 days, and 180 days. So if you'll click on these button, you'll be redirected to the payment page, and we are, providing scholarship to low income countries. So you can reach out to us at marketing@omicslogic.com, or you can directly connect with us at uh, +919876134120. This is the official contact number, and it's active on WhatsApp. Maybe Shri Gauri or Shubham can share the details in the chat, so that those whosoever is interested, they can directly ping us or mail us at marketing@omicslogic.com. So one thing I would like to clear here: we have different prices for different communities. For example, there are pricing there is different pricing for Indian community, that's in INR. There is different pricing for African community, that's in Naira. Then for USA it's different, and so on. So for that, if it's possible, Shri Gauri, could you please share the Learn portal link in the chat box, and those who are interested, they can take a look. So this was, yeah, so this was all about the payment checkout, the research sources, the outcomes, the program structure, etc. And if anyone is having any doubt regarding specialization tracks or the enrollment process, etc., please feel free to ping in the chat box, and I'll be there to guide and assist you. Thank you so much, and over to you, Shri Gauri.
All right. Thank you so much, Sparsh. Let me share the page for enrolling in the chat box. All right. I've shared the portal link for registering for the program. So, does the participants have any queries? One participant has expressed their interest in participating for the poster presentation. Sparsh.
Perfect. So could you please share your email address in the chat box? We'll be sending you the mail. Okay, so you have already shared. Sure, vjsharma152@gmail.com. We'll be sharing the necessary details with you over your mail address. Thank you for sharing.
Thank you so much. So we'll pause for a couple of seconds for the participants to share their queries. And if you have no queries, we'll be ending in just a couple of minutes. All right, so we do have one query. What is the scope of NGS in bioinformatics? All right, OGSP has shared that. Russell is requesting for fellowship scholarship.
Russell, I guess one of our representative, Shubham Kumar, is already in touch with you. So Shubham Kumar will definitely get back to you and will clarify all your doubts if you have any. Thank you so much.
All right, we have another query from Sanzida. Is it really possible to publish an original paper?
Yes, Sanjida, it is possible to publish an original paper, but obviously it takes time. because when it comes to publishing a paper, there are a few protocols which we need to follow. And like obviously you cannot you cannot publish a research project which is being completed in 45 or 60 days. At least you require 3 months or 6 months of project work, and then a full-fledged good manuscript. And then there is a okay, so Ojasvi has already answered yes, it is possible. You have just to find some more key findings from an analysis which is not there in the author's original work. Thank you, Ojasvi.
All right. I hope that answers your query, Sanjita. She wants to know who will help her or will collaborate with her.
So our expert mentors will help her out and collaborate with her. So Sanjita, you can mail us and we can maybe meet one-on-one on a separate Zoom call and clarify all your doubts there.
All right. Thank you so much.
You're
Sanjita, could you please share your email ID as a personal chat so that we can reach out? And we have another query. I have worked on identification of candidate genes in melanoma skin cancer using differential gene expression and co-expression network analysis. And how can I get the ontology of this?
So I guess I have to unmute myself. So we have different pipelines on the server, which gives the results for both KEGG and GEO analysis. Also on NCBI, I guess, and we have that pipeline build if the data set is with the analysis of the GEC and GEO based enrichment analysis. So on the server, we have those pipelines